Mansi Janda. Let's talk about the topic double fertilization in flowering plants. जैसा कि आप सभी जानते हैं फ्लावरिंग प्लांट्स के अंदर हमारे पास बेसिकली दो टाइप के गैमेट्स होते हैं. वन इज मेल गैमेट एंड द सेकंड वन इज फीमेल गैमेट. नाउ द मेल गैमेट इज stamen and the female gamete is pistil, and the female gamete is pistil, or this pistil is also known as the carpel. Now this carpel have further three parts. One is stigma, second one is style, and third one is ovary. Now this male part stamen, this is also divided into the two parts, which is anther and second one is filament. We are going to study about the fertilization. In the process of fertilization, there is a fusion of male gamete and the female gamete. Now the male gametophyte, that is represented by the pollen grain, and the female gametophyte is represented by the egg or the ovum. Female gamete is represented by egg or ovum. Now there is a fusion of male gamete and female gamete that leads to the fertilization process. Now let's talk about the structure of the embryo sac. This is the structure for embryo sac. Here there are two ends. One is chalazal end, second one is micropylar end. At the chalazal end there are three antipodals present, while at the micropylar end there are two synergids present, and one axil is adjacent to that of synergids. There is larger cell which is also known as the central cell. In this central cell there are two polar nuclei present at the mid of the central cell. Now the process of fertilization takes place. Firstly, the pollen grain reach up to the stigma. This is the structure for anther, and this is long stalk. This is known as filament. And this one is anther. Now the pollen grains are produced within this anther, and then the pollen grain reach up to the stigma, and then it leads to the style, and then it enters into the ovary part. In this ovary part there is presence of embryo sac, which is also known as ovule. This is the structure for the embryo sac or ovule. This has two ends. Now the process of pollination takes place. After this pollination, the pollen tube will enter from stigma to style, and then it reaches up to the ovary. In the ovary it reaches up to the ovule sac, and it will lead to the micropylar end. In the micropylar end there are filiform apparatus. These sac-like structures, these are known as filiform apparatus, and these act as the guard cells which guard the entry of the pollen tube into the synergids. Now this pollen tube carry the two male gametes, and it releases the two male gametes into this synergid part, and then this one of the male gamete reaches up to the egg cell, and this one male gamete fuse with the egg cell, and this process is known as fertilization, that will lead in the formation of zygote. Now this zygote further develops into the embryo. Second male gamete then enters into the central cell, and then it fuses with the polar nuclei. Then it fuses with polar nuclei present in the central cell. Hence there is a fusion of three nuclei takes place. This phenomena is known as triple fusion, and the result of the triple fusion is primary endosperm nuclei, and this primary endosperm nuclei form. Then after, the central cell will get converted into the primary endosperm cell. PEC stands for the primary endosperm cell, and PEN stands for the primary endosperm nuclei. This primary endosperm cell further develop into the endosperm, and it will give nourishment to the developing embryo cells. Hence there are two phenomena takes place. One, this is known as fertilization, and the second one is known as triple fusion. So there are two type of fusions takes place. One is syngamy and the second one is triple fusion. So this phenomena is known as double fertilization. This is all about the double fertilization in flowering plants, and the result of double fertilization is one is zygote and the second one is primary endosperm cell. So this is about our double fertilization. Thank you.